So I wish I could speak as well as Ken Ham, but he is the one who inspired me to do this. He was sort of the original founder. And he has all kinds of videos like that if anybody's interested. So, back to your lesser speaker here. So my goals for this course, and if you didn't get a handout in the back, please feel free to grab one on the way out. It has my full syllabus of what I want to talk about each week when we're here. So if there's a topic you're particularly interested in, you make sure you're here for that time. I am still doing the last couple, so don't hold me to exactly the last two presentations there. I'm still kind of working out the details of that. We'll see how this first part goes and we'll go from there. But here's what I want to cover in this course over the next few weeks. So number one, I want to explain the fundamentals of evolution. You can't really attack a problem unless you fully understand it. So I am going to go through sort of the basics of evolution, and that's actually something I'm going to do tonight. And then over the next couple of weeks, we're going to talk about why that theory is so flawed from a scientific standpoint. And then we're going to move on and discuss the problems with the Big Bang Theory. We'll go over what the Big Bang Theory was. It was actually kind of interesting for me to learn a little bit about that. But we're going to explain the evidence for a young Earth, to defend my position that the Earth is 6,000 years old. I've got a whole week dedicated to radioisotope dating, so if that's of interest of yours, it's a little deep. It took me a long time to put that one together, but we're going to talk about radioisotope dating because that is something that is often used to attack Christians. We'll talk about its potential flaws, its potential uses in discussing the age of the Earth. Very interesting group of Christians that have done some great work on this. Then I'm going to review some paleontology. We're going to go through the fossil record. And then a couple weeks discussing the importance of and evidence for Noah's flood. That's actually a very interesting thing. And then the last session, I want to talk about some other frequently asked questions that you may get challenged about. Talk about theistic evolution and compromise theories and whatnot. And we'll kind of wrap up the course with all of that. So, a couple minutes here on the technical nature of this course, and I apologize in advance. Much of what I plan to cover can get pretty technical. And even most of my background is in biology, evolution, so as I got into radioisotope dating and astronomy and Big Bang, I actually had to learn a great deal to put the course together. So it does get pretty technical at times. I've tried really, really hard to simplify this as much as I can, but feel free to ask questions. I tend to talk very fast, as some people who work with me will remind me. But please feel free to ask questions if you don't understand. Also on the back of the syllabus is my email. Feel free to email me. I'll be around afterwards. Because I talk so fast, I typically should have a little extra time at the end for plenty of questions. So bear with me if I end a little early. I apologize. We can chat. You may also want to consider picking up any book on the topic. You can use the ones that I've listed there, or you can use any other. As I say, Answers in Genesis has amazing online resources. You can almost just type in a question and they've got a topic on it, or they've got somebody who's written something about it. Also, the Institute for Creation Research, I'm not as familiar with these folks. Their work is also excellent. Theirs is a little more technical. So if you need a little more technical explanation, I would check out the Institute for Creation Research. So I've tried to call out technical terms in the bold, underlined, highlighted, etc. throughout the presentation. Now I want to talk to you about why, though. Why have this discussion at a church? Why have all this technical discussion? And I think there's three reasons for this, and I think they're all equally important. So first of all, this stuff is technical. So I really appreciate this as a physician, actually. As I went through med school and learned about the wonders and elegance of God's creation. They are essentially boundless. And we are only scratching at the surface in the science world to understanding this. But the more you appreciate how technical some of this stuff is, I think the more you will appreciate the elegance of what God created. And I hope that by understanding some of these technical principles, you will gain an appreciation for the brilliance of the Creator. I'm reminded almost every day of how brilliant the Creator was as I work in cardiology. Now, many of these processes far exceed human understanding, and they always will. As I say, we're merely scratching at the surface. But I hope that you, like me, will find the study of these technical aspects gives you more of a certainty in God's existence and His creation, even if you don't follow every single thing that I say. Second, I want you to understand creationists are scientists too. We're often told to, quote-unquote, trust science over religion. Well, are all the scientists therefore secularist, God-denying heathens? Well, no. And many are not purposely adversarial to God or Christianity. And there are numerous Bible-believing scientists with all kinds of fancy letters after their name. And we're going to explore some of their work as they've written very elegantly about creation. And I would say that their work is on par, honestly, if not superior, as I read some of it, to much of the work done by quote-unquote regular scientists. And we'll try to see that as we get into the explanations, particularly on radioisotope dating. And ultimately, I want you to know that quote-unquote real science can and does support the Genesis account of creation. We'll talk some about that. Finally, I want everyone in this room to have confidence in your faith. So even if you don't grasp everything that I tell you, you're still going to be ahead of 99% of the people out there. The vast, vast majority of people you meet on the street have never thought about these concepts. They've never studied them. They were simply told the Earth is billions of years old, that evolution is there, the Big Bang is there, and they don't really know why. When I tell people I don't believe in evolution, especially if they know my background, I get very blank stares. And it's like I told them I don't believe in gravity. And I actually enjoy that. It warms my heart. Because I love kind of blowing their minds. And honestly, it doesn't take me very long to explain just a couple of the things I'm going to cover over the next several weeks to really get them to start to either question or to just walk away. I don't want you to feel like you have to leave this course and start arguing with everyone. In fact, please don't do that. I actually go out of my way not to do that. I probably was more aggressive with that in my younger years. I've matured, I've got kids of my own. Rather, I want you to feel confident inside that what you know in your heart to be true is true and is backed up by a great deal of quote-unquote science. You don't have to be a scientist to understand that there is science behind this. And so hopefully some next time someone calls you a science denier, you can just retain one or two of these explanations to challenge this course. You might be surprised by what the result. All right. Before I move on to the basics of evolution, does anybody have any questions about the purpose or the goals or where I'm coming from or anything like that? All right. Cool. I guess we're all on the same page. Okay, guys. So, as expected, I'm a little ahead of schedule, so we'll blab some at the end. But so what I want to cover here is I want to go over what you probably were all taught a little bit younger about how evolution works. Before I can really explain evolution, though, I've got to explain some pretty fundamental science. Evolution just means change over time. Things evolve. This theory originated with Darwin's book in 1859 that we've all heard of, On the Origin of Species. Now, interestingly, and this was something I learned too, do you guys know where Darwin's buried since it's written on my slide? So Darwin's buried in a church, which is really ironic. The good news is he's buried in the floor of the church, and so you can go step on his grave. And I have done that. In fact, my whole family made a point of doing that when we were in England. So he originally came out with the original theory that we're all familiar with. And over time, obviously, as questions have come up about it, attempts have been made to fill in some of the gaps of what he originally taught, but his premise remains the same. That basically, from goo to you by way of the zoo, that we slowly over time evolved into more advanced creatures. Now, it is very, very important, and this was a concept that took me a long time in college to understand, but to separate in your mind what I call microevolution, which is change within the species, from macroevolution, which is change into or between species. And we'll talk more about those differences. So what is microevolution? So these are changes achieved within a species by single gene mutations. This will explain things like breeds of dogs, antibiotic resistance. Well, what's the key? You start as a dog, you end as a dog. You start as a bacteria, you end as a bacteria. Microevolution is easily observable. We see it all the time, readily accepted by creationists. Do not let somebody convince you that macroevolution occurred just because microevolution is out there. We hear all the time about the stupid beaks on the stupid finches. Okay? They started as finches, they ended as finches, they've got different looking beaks. Pit bulls and chihuahuas are dogs. They look a heck of a lot different than the beaks do. And we all can readily accept that they are all dogs. These are all finches. This is what microevolution is. Now, this is where I start to get a little technical, so bear with me. This is what I originally got my degree in, so I'm gonna, and you can literally get a PhD in what I'm gonna try to treat you in a few minutes. So bear with me a little bit here, but we're gonna go over basic cellular biology so you understand the fundamentals of evolution. So we've already all heard of DNA. This is the fundamental building block of life. It codes for all the properties of an organism. Everything that makes you, you is in your DNA. We've all heard that. Everything that makes an animal is in their DNA. The way we convert this code into the creation of cells and living organisms is incredibly, incredibly complex. And I don't think anybody fully understands it. But as I say, you can get a PhD in this, so bear with me. So you know where I'm going with this. The big idea is that you start with DNA, you turn that into what's called RNA, to turn that into protein, which forms organs, which come together to form an organism. So I want to work backwards a little bit here, starting with humans. Humans are made of organs. Organs are things like your lungs, your heart, your liver, your kidneys. These organs are made by cells that all work together toward a common goal. The nephrons that make up your kidney are all working together to be kidneys. The myocytes that make up your heart are all working together to be your heart. The activities of these cells that make up the organ are determined by what type of proteins or enzymes is the technical term for this that it produces. So a cell produces enzymes to make certain things happen. That certain thing we make happen, the enzyme catalyzes that reaction. In other words, it greatly speeds up an interaction between two substances to produce a new substance. So for anybody lactose intolerant, you don't have to admit anything to yourself, but there is an enzyme called lactase that causes the breakdown of lactose into two other components the cell can use more easily. Lactase is an example of an enzyme. It catalyzes the breakdown of a complex molecule, lactose, into simpler molecules. That's an example of a protein and an enzyme. Other proteins in your body include things like antibodies, insulin, hemoglobin, albumin, things you probably have all heard of or at least seen on your lab results if you print copies. So what are proteins? Well, proteins are composed of subblocks called amino acids. There are 20 amino acids. The number and the order of these amino acids determine the protein that is made. Interestingly, we as humans can only make 11 amino acids. Anybody heard of essential amino acids? Those are things you have to get from your diet. Your body can only make 11 of these. So you have to consume the other 9 amino acids. That's why when you tell people eat more protein, that's kind of what we're getting at here. You got to get those amino acids from somewhere. So this is an interesting side note on veganism. Sometimes it's a little harder to get those amino acids. So if you ever hear the term essential amino acids, or if any of you are strict vegans, pay careful attention to making sure you're getting those. So the average protein is made of somewhere between 375 and 400 amino acids, but they can be much longer and maybe a little bit shorter. The order of these amino acids is what is coded for in DNA. So when you get the amino acids all put together, and down here, these are abbreviations for various amino acids. They start by just being put in order. We call this the primary structure. And then through very complex mechanisms that you can take a whole semester-long course in microbiology or biochemistry if you're interested, you can learn how they fold into what are called alpha helices and beta sheets. And then these complex structures fold on top of each other into what we call a tertiary structure, and then it folds again to the quaternary. I'm not going to deal with all the details, but I do want you to appreciate the elegance and the complexity of this. That this is not something that just simply happens. Turning back to DNA. DNA exists ultimately in what are called base pairs. And you may have heard of some of these before, but DNA is only made of 4 molecules. Well, it's made of a lot of molecules, but the important molecules are this A, T, G, and C. Those are the only 4 letters that you get in your DNA. And so A binds with T and G binds with C. And you're probably familiar with this structure where you see the DNA binding to each other or attaching to each other in this fashion. And then these pairs are wrapped in what is called the double helix, which was discovered in 1953. They won the Nobel Prize for that. And then these, and I'm going to show you a picture of this, because this is something else I want you to appreciate the elegance of. These double helices are folded over many, 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 many times to form what are called chromosomes that you probably have heard of. And humans have 23 pairs of chromosomes. You get one set from each parent. Now, each cell, cells are microscopic, each cell contains 6 feet worth of DNA. Okay? Each human has 10 billion miles of DNA in them. Okay? So if you're going to wrap that much DNA, something that small that we can't see, that is really, really, really tight wrapping. Much better than my wife can do with presents. Just so you appreciate the elegance of this, these are those base pairs that we talked about. This is the double helix. The double helix then starts to wrap around these other proteins called histones. Don't worry about the term. But these histones, you see, then fold back on themselves into this very complex structure called a chromosome. And then they fold up more and 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 more until you get what you might have seen before, these pictures of chromosomes. These are incredibly, incredibly tightly wrapped. It is a miracle in and of itself, or what I would prefer to believe, the genius of God, that designed a way for all of this wrapping to occur. So, DNA is then unwound. I'm going to skip all of the stuff that goes into unwinding, because that is a very, very complex explanation. Again, probably a semester's worth of discussion. But suffice it to say that DNA is unwound at certain points that are predestined and then transcribed. The term is transcribed into RNA. Specifically something called messenger RNA. So the DNA takes its code and using other enzymes turns the DNA into RNA, which is now single-stranded. It's not double-wrapped like this. So now this RNA contains a mirror copy of the gene encoded by DNA. If you see the letter U, don't worry about it. RNA uses uracil instead of thymine. It's a minor point, don't worry about it. But essentially the letters are the same, but we got a mirror copy here. So how do we turn that into proteins? Well, this messenger RNA, so we've transcribed DNA into RNA. Messenger RNA is now translated into protein through the use of something called ribosomes. And this is very elegant how this works, but each gene base pair, so each set of 3 letters that we got coded from the DNA, which turned into RNA, each 3 letters is called a codon. And each of these codons tells the cell which amino acid to put there. Miracle of evolution. Transfer RNA carries that amino acid, matches up with the same way we know things matches up, and pulls in its amino acid. And so then the next one comes in, and that amino acid is attached. And the next one comes in, and that amino acid is attached. And slowly we attach these amino acids one after one after one into what is called a polypeptide, because we always have to have fancy terms. And this eventually turns into a protein. So the collection of amino acids becomes a protein, which then has to go through the folding. So again, DNA turns into RNA, which codes for these proteins, which are your genes. This is simply the process in pictures. We're turning the, did I already lose my laser? Oh, it's white, that way I'll show. So the DNA through RNA polymerase turns it into RNA. This ribosome comes in and builds the protein that comes out. Now keep in mind, I didn't put this in my slides, keep in mind, this is happening in milliseconds. This is happening over and over and over in each one of your cells all the time. Every time a cell divides, every time a hair grows, every time your heart beats, every time you're doing anything, you're doing this over and over and over. And this all happens incredibly fast and incredibly rapidly. So let me talk about these codons. I told you there's 3-letter codes. The magic code called AUG, which is the universal start codon. All proteins start with that letter, or with that code, excuse me. So they all code for the same amino acid. Then there are 3 that say stop. So there's one that says start, and it goes along until it runs into one of these 3. And keep in mind this idea of a stop codon, because this is going to come up in a minute. But basically, your protein is built, it starts here, and it keeps going every 3 letters until it runs into one of these. Now, all the other letter combinations code for a certain amino acid, and you can pull up books that'll tell you exactly. So if you've got U, that's phenylalanine. If I can't remember, methionine, I think. So if you've got AUG, which is that start codon, every protein in your body starts with methionine. Okay. So this is how the gene code works, how DNA is translated into protein. So, was that? Go ahead, question.
No, I just said, did you guys get that?
Oh, okay. Yeah, all right. So now let's talk about mutations, the mechanism of evolution. So errors in the genetic code can be all kinds of things. You can get an insertion and throw in an extra base pair, a deletion, one comes out, or you change one letter into another, and you do this in the DNA, and it happens when the DNA is copying itself. And then the copy is copied, and it's copied and copied and copied. So a deletion or insertion changes, when you pull out one letter, think of it, you're reading a sentence and you pulled one letter out of the sentence. Now you got to recombine it. It makes no sense whatsoever. You were reading by 3s. Think of it by counting by 3s. 1, 2, 3, 4, 5, 6. Suddenly you pull out the 3. 1, 2, 4, 5, 6, 7. 8, 9, 10. So you changed your counting completely. You get a completely different protein because your counting by 3s has been screwed up. And this is called a frame shift mutation. A substitution is much easier to understand. You change one for another. A G becomes an A. And you get a different amino acid in there. So you change one amino acid. A missense mutation is that. You changed one amino acid. A nonsense mutation, remember I told you what the stop codons are? Well, what happens if you change into a stop codon? Well, now your amino acid has changed to a stop codon, you won't produce that protein anymore. So if you were supposed to produce a protein that was 300 long, and amino acid number 2 turns into a stop codon, you don't produce your protein. So let's talk about how this works in microevolution. So bacteria are very useful for studying microevolution because they produce very, very quickly. E. coli, which you probably all heard of, reproduce every 20 minutes. And they do so purely by forming copies of themselves, so they're very easy to study. On top of that, bacteria has some very interesting properties about it in that they are very prone to mutation by the 3 things I listed here in bold: transformation, transduction, and conjugation. Suffice it to say, without going into a great deal of detail, these are ways for a bacteria to pick up new DNA. When bacteria are floating around and they get destroyed, some of their DNA just releases into the medium. Other bacteria can pick that up and incorporate that DNA. Viruses can transfer DNA from one bacteria to another. And sometimes bacteria mate, in a way, and one bacteria donates DNA to another. So these are things humans and more advanced organisms do not do, but suffice it to say, all of these things make bacteria very useful for studying evolution. And this is why antibiotic resistance is so prevalent. There are so many different ways for bacteria to mutate. So let me give you an example of natural selection. I've got 1 billion bacteria. Okay? By the way, which probably fit on my thumbnail. And I exposed them all to penicillin. And the vast majority, 990,000 of those bacteria die. However, 10,000, which is a small percentage, 0.001% live. Why? Well, through those methods I described before, they upregulated, meaning they produced more genes, or even acquired other genes that made them resistant to penicillin. These are things like antitoxins. Antitoxins are things bacteria produce that destroys penicillin. You take penicillin, and the bacteria produces something that destroys penicillin. Penicillin actually attacks the way the cell forms. they can change their model of their cells so that they're resistant to that. So what happens now? Well, those remaining bacteria, those 10,000 that survive, they can go on living and reproducing. And they quickly build the colony back up to 1 billion. And so the next time penicillin is introduced, only 900,000 die. And new mutations occur. And the colony gets stronger and stronger until the colony is completely resistant. This is the process of natural selection. We have selected out the weaker and held on to the stronger. Something else many of you have probably seen in your textbooks are the moths. Prior to the Industrial Revolution in England, people in London noticed numerous white moths, and that they disappeared. And the peppered moths became more prominent. So the peppered moths and the dark moths. Well, why? The theory is that before the Industrial Revolution, the white moths could hide easily on all of the buildings. But as the Industrial Revolution occurred, soot covered all the buildings and made the dark moths disguise themselves much better. And so the white moths got eaten, and the dark moths hid. And so this was a classic example of natural selection because the birds came and wiped out the white moths. Same thing you've seen in giraffes, right? If only I could reach that fruit just a little bit higher. The original short-necked ancestor becomes a little longer, and a little longer, and a little longer, until we have wiped out the short giraffe, and now the long giraffe is the only one that can survive because apparently food only grows it that way. Now, the theory of macroevolution is that each of these processes we've discussed in natural selection were dictated by a single gene mutation that somehow gave the organism a survival advantage, allowed it to live better, longer, or more efficiently, and pass on its advantageous trait. So there were a bunch of tiny giraffes. The food apparently ran out at 4'1", and a 4'2" giraffe dominated them all, while the 4'1" died. And the 4'2" guy went on to pass on his genes. So a couple generations later, the food grew to 5 feet. So only the 5 feet giraffe lived. Okay? So what we're taught, science, in the lesser organisms eventually died off because it was disadvantaged, and environmental pressures like changing weather, new predators, changing soot, drove these changes and forced these processes. And what are you told over and over and over? For us, these things just take time. These are slow changes, massive amounts of time is involved here. So, these are the basics of evolution. Modern evolutionary theory purports to explain all living organisms as products of microevolution, transitioning to macroevolution through the process of natural selection. They prefer that term rather than from breeding, by way of disease. So we started with single-celled organisms, transitioned to multicellular organisms, eventually became complex animals, and then modern animals. Next week, I want to fundamentally dissect each of these various premises and discuss why each one is so flawed. So, about all I have to say on that. So we do have a couple of minutes before things are over. If anybody has any broad questions, feel free to shoot them out now. If you want to talk to me afterwards, I'm happy to do that. You can also email me. Anybody have any? Yes, sir.
I missed one of the 7 C's.
Yes.
That slide item was creation, corruption, catastrophe, and this the 4th one.
It's the Tower of Babylon, but I can't remember what the C is. Confusion, that's right. Confusion of the language. Yeah, so Tower of Babylon, Tower of Babylon. Yeah, that's Answers in Genesis. That's they have their 7 C's. They started with the Creation Museum to kind of cover creation, and then they talked about catastrophe, which is why they built the ark. The thing they are working on right now actually is building a life-size Tower of Babel, not to get destroyed and separated, but to make the point of the importance of the Genesis story that way. Yes, sir.
Hey, is there anybody out there who's doing any work or proposing any hypotheses for how long microevolution then takes if we can try to explain like all the variances in dogs.
Sure. So it depends on the species, right? Microevolution in a bacteria is 20 minutes or a couple of hours. I mean, we used to do these kinds of experiments in college. You'd actually program so you could reproduce antibiotic-resistant bacteria. It's pretty society. But when you're talking about dogs and whatnot, yeah, it takes a little longer. And as we get to the ark and some of the what we think God means by kinds and how speciation occurs, we'll talk a little more about that. I don't know off the top of my head how long each thing takes. My assumption would be you're talking a few generations. So if it's dogs, they take what, a year or two once they're grown to produce. So you're probably looking at 20, 30, 40 years of continuing down that pathway. So you're probably looking at several generations before you get a change. But bacteria can happen very quickly. Well, guys, I appreciate everybody coming. I'm really flattered to have so many here. I hope a few of you will come next week. Hope I didn't bore anybody. And if you have additional questions, feel free to come hit me up, okay? And if you didn't get a copy of the syllabus or the books, they're all in the back, and they'll tell you what topics we're talking about. If you need any additional recommendations,